be moving back to genetics and talk a bit about how genetic variation affects chromatin states and mediates response to drug. And this is work done in mice. And mice are really great for this sort of basic research because you can control the population. And so here I'm showing the results from an eQTL study, and this is where I'll start. This is livers taken from mice from an 8-way intercross. So we started with 8 inbred mouse strains, all had been sequenced, and we mixed up their genomes and outbred them, and then did eQTL mapping on about 480 livers is what you're seeing here. So you can see there's a very strong local eQTL line right down the middle, and then some stuff off the diagonal. Oops. And so we are interested in modeling these and trying to understand how different alleles within this population are influencing the expression. And so we did eQTL analysis, and you can see here's one gene on chromosome 6. We're mapping it to its local region. You can see there's a very strong eQTL there. That's a log score of about 140, so essentially a p-value of 0. And it's a very strong QTL that's really driven by this green strain here, this castaneous strain, reducing the expression of that transcript very strongly. And so basically what we do when we have these haplotypes, because we can reconstruct from any one of these heterozygous diploid genomes, the ancestry of every allele, and so we can regress on the allele. So instead of doing SNP regressions here like you commonly do in GWAS, we can actually do haplotype regressions, and that's what you're seeing here. And so we can find which haplotypes have which effects. So the green strain castaneous has a negative effect on the expression. The red strain PWK sort of is intermediate, and all the others are fairly high. So there's probably some variance in Cas driving expression down, other or the same in PWK, but not as much, and then these others are probably about the same. Okay, so we can map this everywhere in the genome. We find thousands of eQTL, both in cis and in trans, and we can go into the inbred mice. So these are the founder strains that we mixed up, and look at the expression of this gene again in the liver, and you can see that what we often see is that the pattern that you see in the inbred strain matches what you see here, which is basically to say castaneous has the lowest expression. It's the one that drives down when you randomize the genetic background. So in many cases, what you see in the inbreds is the same as if you mixed up all of the background, so you know that's really a cis effect. Okay, so the next question was, what might be mediating these effects, and how much of this might be epigenetically driven? And so, as was talked about in earlier talks, you can have a SNP that somehow changes the chromatin state in the promoter or an enhancer for a given gene. And so that cis eQTL would be really chromatin dependent, probably through some remodeling factor. Binding to the SNP, you would be changing the chromatin state. Or you could have cases where the SNP was acting in a totally chromatin independent way. Say you had a transcription factor that would bind that SNP and all you needed was open chromatin there, and it actually wasn't changing the chromatin level at all. Or you could have SNPs throughout the gene which could change its degradation rate or its splice variation or any such thing. Okay, so there are two possibilities here, chromatin dependent, chromatin independent, and we wanted to estimate really how much of these were chromatin dependent, and also think about this in the context of drug treatment as well, because when we're thinking about these individual SNPs affecting a response, affecting the functioning of an organ like the liver, generally we're thinking about how will this relate to personalized medicine and something like personalized drug response. So we didn't have the resources to run all of these ChIP-seq assays on all 480 of these mice. So instead, we just went to the inbred founders and used those as proxies. Because all of these are cis eQTL, we were taking as a first approximation, the inbreds would represent those sort of epihaplotypes. Okay, so we took 9 inbred strains, the 8 founders that I just discussed, as well as DBA, which is important for other reasons. We isolated hepatocytes, so purified hepatocytes from each of these mice, and we had all 9 untreated, and we treated 5 of them with dexamethasone, which is a very strong glucocorticoid, which should have very strong activity in the liver. And we took 3 female replicates from each mouse, and with the pool of hepatocytes we get from each mouse, we ran all these assays. So we did RNA-seq, bisulfite-seq for DNA methylation, and then a number of ChIP-seq assays to look at chromatin states. And so I'm going to talk about the ChIP-seq today. We haven't finished with the the bisulfite yet. Okay, so we can call peaks, like we've all seen. Here's a region for one of the strains. We're calling peaks for H3K4 trimethylation, H3K4 monomethylation, and H3K27 trimethylation throughout the genome. We use MAX, pretty standard peak calling. You'll notice that some of the 27 peaks are rather broad and a bit more difficult to call. That shows up later in the work, especially here where we see we have far fewer K27 peaks called across strains. We have about 40,000 H3K4 trimethylation peaks and somewhere in the neighborhood of 90,000 H3K4 monomethylation peaks per strain. So these are all of the inbred strains across here, and they have their requisite color coding. If you look at consensus peaks that appear in one or more strains, you see that many of these are shared. They're not, all of these peaks aren't shared across all of the strains, but many are shared. And not surprisingly, many of the H3K4 peaks overlap genes, and not so many of the 27 trimethylation peaks, which is an insulator, overlap genes. Furthermore, if you ask, doing a strain comparison of the untreated samples, how much correlation do you see between a peak and its nearest transcript levels? You have positive correlation for the H3K4 modifications as expected. Those are pro-regulatory peaks that increase expression, whereas you have negative correlation for the H3K27 trimethylation peak. As a repressor, that's what you expect as well. Okay, so this all seems to be working. So we went ahead and organized all of these peaks into what we're calling basically the peakome for all these mouse strains. So we line up all the peaks, and if a peak appears in any strain, we assess it in every strain, whether it was significant or not. So we're still putting the quantification in, and ran just basic analysis like PCA. So here are PCA plots for the RNA, standard transcriptome, and then 3 of the marks. You can see a major separation here by strain. So these are only the strains which we've treated with glucocorticoids. So closed circles are no treatment, open circles are the treated with dexamethasone samples. So you can see everything is separating in the first 2 principal components by strain, and you're getting a group of strains here, another strain here, and another here, and this is really just mouse phylogeny. So these are all Mus musculus domesticus substrains. This is a Castanea substrain, and this is a Mus musculus musculus substrain. Okay, so that's to be expected, that separation, and we see essentially the exact same separation in K4 monomethylation and trimethylation. K27 trimethylation looks a bit different. You still see the separation, but you're also getting more separation between the domesticus strains. And there's just less genetic variation in that mark. And so you're not seeing the extremes that you see with those 2 outlier strains in that assay. Incidentally, all of the other domesticus strains that we didn't assay with dexamethasone pile up here as well. The other thing you'll notice is that the separation really is genetic here, and that within these clusters you can see the treated and untreateds in every case, which is to say that the genetic variation is driving far more variation in all of these assays than what is really a very strong perturbation of the liver with a steroid. Okay. And you actually have to get to principal component 5 before you see a clear, consistent separation between treated and untreated strains. So the lesson here is genetic variation can really overwhelm even the effect of a drug. And again, these are mice with quite a bit of variation within the strains that we mixed, and perhaps in, say, a human population, you're not going to see that much variation, but it's still quite a lesson about the power of genetics. If we do look at the response to dexamethasone and just isolate strain by strain how many differentially expressed genes and or peaks we see, you can see that it varies by strain. So the number of transcripts that we see different varies from this PWK strain in red, which is over almost 2,000 transcripts, all the way down to this WSB strain, which is barely responding at all. It's only a couple hundred transcripts. And some are up and some are down. And we see pretty consistent results across these regulatory marks as well, with the PWK very high and the WSB very low. Okay, so the WSB strain, whatever its biology is, it's just not really responding to the steroid treatment. Not surprisingly, the genes that are up have increased expression, as well as the peaks that are up are near genes that are steroid metabolism-related genes. This is the liver. This is where steroids are metabolized. And the downregulated genes tend to be immune-related genes. These steroids are immunosuppressors, so that's not surprising either. If we look at the peaks that show differential expression or differential levels across these strains and put them all together, you'll see we have about 8,000 K4 trimethylation peaks, 13,000 K4 monomethylation, and about 1,000 with both. the vast majority, especially these overlapping peaks, have a glucocorticoid receptor binding motif, and that's shown here. So the glucocorticoid receptor directly binds the drug, is imported into the nucleus, and activates or suppresses genes. And so that's mostly what we're seeing here. Everything, as far as we can, or the majority of effects, as far as we can tell here, are being mediated by this glucocorticoid receptor activity. In terms of the genetic variation, so moving away from the treatment, just looking at the non-treated strains and genetic variation, the strain-to-strain differences in the genes that are different vary quite a bit by gene function. So housekeeping genes and highly conserved genes don't vary all that much in terms of log 2-fold change here. So less than a factor of 2-fold change, which makes sense, as housekeeping genes probably wouldn't vary all that much. They're pretty stable. They're required for all of the strains, whereas strains that are more liver-specific tend to show more variation. Okay, so these functionally specific genes tend to be the ones that allow genetic variation to perturb them. And then many, of course, unclassified RNAs are spread across the entire range. And so many of these perhaps are also relevant to the phenotype differences between the strains. Okay, so the last thing we did was to really put these marks together and try to understand how different chromatin states might be related to differential expression. And so we basically, what I call, encoded the data. We used chromHMM to call genome annotations in terms of chromatin state for every one of these strains individually. Okay, so here are all 9 inbred strains. This is the IGF-1 locus, so that's insulin growth factor 1, a very important gene, highly expressed in the liver, highly expressed in all of these strains, and doesn't really show any variation in terms of gene expression. And it doesn't really show much variation in terms of these epigenetic annotations either. So in every case, you have an active promoter there in purple. You have massive strong enhancer signal all around the gene, especially around the start site. You could call those super enhancers that stretch across the site if you want. There are also a number of enhancer sites upstream, and these tend to show up in the majority of strains. And then you have these repressor sites in these intergenic regions, which we see as a common feature as well. Okay, so we can go through every strain and look for genes like this with very similar patterns, but of course the interesting ones are the ones with variation. So here's an example, GABRB3, that has a lot of repressor signal around its start site and only strong active promoters in 2 of the substrains, Cas and PWK. Okay, so your guess might be that this gene is expressed higher in these 2 strains and low in those other strains, and if you look at the RNA data, indeed you would see that. So Cas and PWK are high and the other are low. Some of them are essentially 0. Okay, so that's nice. You see a concordance between the chromatin state and the RNA, and now you can ask, if I go back to that eQTL study in the liver, do I see the same sort of effects? That is to say, if I were to randomize the rest of the genetic background and only look at the local effects, do I see an eQTL that matches this? And this is a case where you do. So here's the LOD score of significance is almost a 100, again driven by a very strong positive effect from this green Castaneous strain. And the secondary is this PWK strain, which is high, and then all the others are sort of down here low. Okay, so you're seeing a match between the mRNA levels, the chromatin organization, and the eQTL. And so this suggests to us that this is an eQTL where there's some variant that is probably changing the chromatin state here, leading to differences in expression. Okay, so we don't know how that's being mediated in terms of which factor is modifying the chromatin, but that's suggestive that we're seeing a concordance, and we have a chromatin hypothesis for this eQTL. And we don't always see that. So here's another transcript to REC8. It has not a lot of activity across any of the strains, but it has a bit more in black 6 and WSB, and sure enough, black 6 is the highest, really the only strain where you see a lot of expression of this gene. And so these are concordant, but if you look at the randomized background, if you look at the eQTL, you don't see that. The black 6 strain is right here in the middle with all the others. And the eQTL, which is significant, is really being driven by the purple strain, WSB, which is sort of higher than the others, but from this data here, we really wouldn't say so. And so this is suggesting that there's actually something else in the black 6 inbred strain background that's causing this. And if you randomize the genetic background, it goes away, which is what we see here. And then we also see cases where the chromatin is discordant with both the expression in the inbred strains and the eQTL. So this is the one I showed in the beginning, PON2. Its transcript is downregulated by having a green or a red allele. It has low green and red expression, especially low in CAST. But if you look across the chromatin states, across all these strains, CAST is down here at the bottom, and it really doesn't look at all different than the others. And so there's no obvious driver of genetic or of transcript variation in the epigenetic marks there. Okay, so that could be due to something else in the CAST transcript causing degradation, or a splice variant, or maybe there's a SNP in this active chromatin here for a transcription factor that cannot bind. Okay, so if we put all this together, I'm plotting here how often we see concordance between the eQTL effects, so this is randomized background local eQTL, and the inbred RNA-seq on the x-axis, and so this is biased to the right because generally speaking you see concordance between a purified hepatocyte from the inbred and a full liver eQTL. And then on the y-axis here it's the eQTL effects and an approximation of the active chromatin within the region. We took the gene plus or minus 1 kb and basically summed up the proportion of chromatin that was in an active state. And so you can see there's a bias toward the top here too, and these, by the way, are the most significant liver eQTL with a lot above 20. If we reduce that, things fill in a similar way. You just can't see because everything goes black. And so we can see that there about a quarter of these eQTL can be explained by mediation through the chromatin state, because we have everything in concordance there. There's a local eQTL, its effects match the chromatin differences, which match the inbred mouse expression. This group, especially down here as you get lower, are showing concordance between the inbred expression and the eQTL in the livers, but not through chromatin. Okay, so those are probably, local eQTL probably being regulated some other way. Okay, so that could be some of the DNA methylation that we haven't got in here yet, or it could be entirely other mechanisms. We have about 500 genes here that really don't correlate. The eQTL doesn't correlate with either of these factors, and those actually could be non-hepatocytes in the liver. So any sort of variation you see in another cell type or an infiltrating cell would not map back onto these hepatocyte purified samples we have, so that's what that could be. And then up here we have the interesting case where the eQTL matches the chromatin, but not the transcript expression in the inbred. So that was like that REC8, where there's probably something else happening in that inbred, perhaps in trans, that gets washed out in cis, and the chromatin matches what you see in cis, but there's something happening in trans in the inbred. Okay, so to summarize, we really sort of surveyed sort of what we get if we think about how genetics affects these molecular epigenetic marks. We see that genetic variation, not surprisingly, leads to strong epigenetic variation that's correlated with transcript variation. The transcriptional response to dexamethasone is really mediated by the background. So when you have different genetic backgrounds, you get very different transcript and chromatin patterns, and they match generally. That response is really driven by glucocorticoid receptor DNA binding. Those are the loci where we see the effects. And finally, doing this sort of epigenetics in this sort of model system allow us to interpret a lot of these eQTL results, and we can guess that about 25% of those local eQTL are probably mediated by effects of the genetic variation on chromatin. Okay, so I'll finish by thanking collaborators as well as folks in my lab. Most of this work was done by Katrina Spruce and Anna Tyler. There's my funding and some contact information if you'd like more information. Thanks.
Thanks. Questions?
How did you map the mouse strains? Because basically the reference genome is black 6. So did you take that into account or just went with it?
So we mapped to individual reference genomes. So we went to the Sanger database, which we had to do twice because they updated with de novo assemblies recently. But for basically every read, we tried to map to all 8 genomes. And
All right.
whichever it mapped the best, that's where we assign the read.
Thank you.
I had a technical question. So the chromatin state assignments are sort of a categorical, discrete assignment. How do you sort of formally do this correlation?
Yeah, so in this case, we just looked across the annotations of the genome around a gene and looked at the percentage of that area or that length that was active state.
Okay.
So it's very rough. We're looking into ways to quantify that better. And of course, that avoids the relevant question of, are there certain regions that are more important than others across that region? And probably there are. We really can't get at that here because we don't actually have the marks in every strain or in every mouse that we did the eQTL analysis in.
Okay, thanks.